Thank you.
So tell us about how you became originally interested in this particular area.
The short version is when I went to graduate school, I was interested in working on the hippocampus, not the retina. And really at that time, physiology was the main trade of the neurobiologist. And there was a lab. I was a graduate student at UCSF, and the lab that I was the most interested in, I won't name his name, but he worked in hippocampus on learning and memory. And when I got there, I went to him first, asked if he was interested in having a student rotate, and he told me the lab was full. So I scrambled, found another lab, which happened to be a retinal physiology lab, and I really didn't know anything about the retina at all, and quite honestly wasn't sure I had any interest in it because I really wanted to learn about memory plus synaptic plasticity. Nevertheless, I did my graduate work there, was fortunate enough to have one of my papers published in Nature, did my postdoctoral fellowship at the Volum Institute, was fortunate enough to get another paper in Nature there, and then moved to the Albert Einstein College of Medicine, all the while staying in the same field. The retina is really a family, I think, and once you become part of the family, it's very difficult to leave. And by difficult, I mean one does not want to leave. So that's sort of the background of how I got started, and I guess it was basically an accident as much as anything.
You're one of the moderators of a major session here at ARVO 2015. Tell us a little bit about that session.
So the session is going to focus on one of the interneurons in the retina called the bipolar cell. And the bipolar cell is a cell that I've been studying I was thinking about how long as I was walking over here, and I think the best estimate I can come up with is about 35 years. So I started working on this cell, its function, what happens to the cell in disease as well as in health when I was a graduate student. And other projects have come into the lab, of course, and have come and gone. But the bipolar cell remains central to the work that we do. And so I was asked to put together a symposium and basically on any subject I wanted to in the retina, and I chose the bipolar cell. And that's the session that I'll be moderating.
How's our understanding of the bipolar cell changed over the last 10 years?
Well, for one thing, we are starting to, the synapse between the photoreceptor and the bipolar cell is very complicated. There are many moving parts to it, lots of different proteins. And we've gone from just a physiological understanding. We started with an understanding that the synapse inverts the signal, if you will, the sign, but we didn't really know how this was done or what proteins were involved. And the last 10 years, I would like to say that almost all of the players have been identified, all of the different proteins, and their role in this transduction process have been identified. And along the way, we've discovered a lot about different families, and I mean human families, who have various mutations in these individual proteins, and how these mutations lead to, among other diseases, night blindness, congenital stationary night blindness. So I think there's been a lot of progress made molecularly and also genetically.
What are the potential clinical implications of the fact that you've now characterized so many of these proteins?
Well, so hopefully in the future with gene therapy, we can go in and actually correct many of these mutations are single point mutations on single genes. And so I think they're very good candidates for gene therapy. And hopefully we'll be able to use this information to restore vision to a lot of people who have this disease.
Are there some proteins that are more important than others in your mind?
So I would say one of the most important proteins, and I'm not being biased, I My lab was one of the labs that discovered or characterized this protein, but I think it's fair to say that one of the most important proteins is the synaptic protein, TRPM1 is its name. And TRPM1, along with a second protein called nyctalopin, whose function we don't completely understand, seem to be the proteins that are the major targets for genetic mutations. And so I would say TRPM1 is probably the most important protein. So TRPM1 is actually forms the synaptic channel. So without this protein, current cannot flow across the membrane, and there's no response in the bipolar cell to light.
Are you aware of which groups are currently working on the genetic solution to the problem with TRPM1?
So Dr. Neil Peachy at the Cleveland Clinic is working on this problem. Dr. Ron Gregg is working on this problem. And there are, around the world, several other groups in Europe and also Asia who are working on this problem. In the United States, I'd say those two labs are the two major labs that are working on the genetics, and I help them by doing some of the electrophysiology on some of these mutants. Of course, we're not doing the electrophysiology on humans, but we've developed a number of mouse models that basically have the same genetic defect as the human. And the TRPM1 channel is very highly conserved between humans and mice. So a mouse model, I think, here is very appropriate.
Well, that's excellent news. Which of these programs appears to be the most advanced in your mind? I know it's a tough question, but if you were to rank them.
I would say Dr. Peachy's program is probably the highest ranked. Is that going to be on part?
No. So, changing gears a little.
Sure.
You've obviously done a wide spectrum of research. What are some of the other areas that you've been involved in and are continuing to look at going forward?
So there's two general areas. Of course, bipolar cells. And one idea that we have to help restore vision to people who have lost their vision due to macular degeneration or other photoreceptor diseases is to insert channels in the bipolar cell that themselves are sensitive to light. And to be honest, the technology for really making this useful for humans is still a few years away. But my lab and several other labs at this meeting are on the forefront of doing that. And the idea simply is to insert genetically using a virus a channel that is able to sense the light, and when it senses the light, the channel opens and current flows through. And so in this way we can sort of hijack the bipolar cell and make it directly sensitive to light. Therefore, the photoreceptors are now unnecessary. So this is one of the projects that we're doing.